Next, we will be covering the leucoplast phenomenon caused by gene MinE1. Leucoplasts are plastids, a family of organelles found in plant cells. They help plants store energy and can form storage components in the plant, such as the potato part of a potato plant. Generally, they are most likely to be spotted in the non-photosynthetic part of the plant, such as the root cells. Another job is to transport macromolecules between cells. Manufacturing cell products like fatty acids and proteins is also common. Leucoplasts themselves are fairly plain. They are essentially chloroplasts without the chlorophyll. Possessing a clear color and no specific shape, they start out as etioplasts and become specialized. If they are exposed to light, they become chloroplasts. If they remain leucoplasts, they might store extra material, like starch-storing amyloplasts, fat-storing alloplasts, or the protein-storing proteinoplasts. They can transport these products by connecting to each other through stromules and provide a pathway, allowing macromolecules as well as other organelles to travel along them. However, now research shows that due to a genetic disorder, the duplication of leucoplasts occurs abnormally. The gene, called MinE1, is one of several genes that helps regulate plastid division. This disorder was found in experiments involving the expression of certain genes in the plant Arabidopsis thaliana, and how they are expressed differently in bacteria and plant cells. The disorder starts with duplication of the gene that regulates plastid division. Typically, a cell will reach a certain size before entering mitosis, or in this case, the plastids. When this gene is overexpressed, the plastids will grow longer instead of dividing and lowers the overall number of leucoplasts per cell. Because there are fewer leucoplasts, stromules are less likely to form and affects the transport of cellular products between cells. Another symptom of this disorder is that instead of forming chains, the mutant plastids will grow in ways that they do not typically grow. For example, if it was supposed to form a two-way fork, it could branch off into 5 or more forks instead. Unfortunately, this is a genetic disorder and has no real treatment. However, it does not affect the plant on a large scale, as it does not eradicate the wild-type function of the leucoplasts. It can be considered a non-harmful mutation, as all it really does is cause enlargement in plastids, though not to the point where they are not able to function. To say the least, this is a lucky mutation in the millions possible for a plant cell.